Okay, so this week is going to be a very short lecture on membrane transport. There's the learning objective for this week. So how do cells transport substances across their membranes? If you remember what the membrane looks like, it's a phospholipid bilayer. So we have phosphate heads and lipid tails. And there's two rows of these, which is why we call it a bilayer. In order for substances to get through this membrane, they have to be lipid-based or nonpolar molecules and small. Not much stuff can just float right through the walls here. Other times, we have special transport proteins, and they allow for substances to move through them. But they're highly selective, which is why this is called a selectively permeable membrane. It chooses which substances can pass through. So for each protein, it only allows certain types of substances to pass through. Now we can have passive transport or active transport. Passive transport does not require energy, while active transport does require energy. And then we have endocytosis and exocytosis, which are examples of bulk transport. So we're going to go through some of these now. So passive transport, again, does not require energy. And our major form of energy, like we talked about earlier, is adenosine triphosphate. Or ATP. So this is not required for passive transport. And what passive transport is doing is it's the movement of substances from an area of high concentration to low concentration. Another word for this, or another phrase for this, is moving a substance down its concentration gradient. Now if you look at this picture, as time goes on, we start with a whole bunch of substances concentrated here. We have a high concentration on one side and a low concentration on the other side. Now as passive transport happens, we have the movement of molecules down their concentration gradient from an area of high concentration to low concentration until we've reached an equilibrium. And an equilibrium is when we have equal concentrations on both sides of the membrane. So once we're in equilibrium, these molecules can freely float back and forth here, but overall we're not going to have a whole bunch of molecules moving in one direction or the other. They're kind of set, they're happy, they're in an equilibrium, equal concentrations on both sides of a membrane. Simple diffusion is a type of passive transport. But usually when we're talking about diffusion, we're talking about the movement from an area of high concentration to low concentration, and we don't have to necessarily be talking about a membrane being present. So if we have a can of soda, so let's say we open up a can of soda. We've popped it open. Now we have carbon dioxide that's super concentrated in here. If we let the can of soda sit out, the carbon dioxide is slowly going to seep out from an area of high concentration to low concentration. But it's not moving through a membrane, it's moving through the open soda can top. So with simple diffusion, it follows along with all of those definitions of passive transport. It may or may not require a membrane. Osmosis is the passive transport of water. So it's the movement of water from an area of high water concentration to low. Or it's the movement of water from an area of low solute concentration to high. And I'm going to demonstrate these two aspects of osmosis here. So if we think about this situation, now we want to create an equilibrium where we have equal concentrations on both sides of this membrane here. So this membrane is dividing these two compartments. Water can flow freely between the two sides. So is water going to move towards side 2 or towards side 1? Well, if we want to equalize the water on both sides, then obviously water is going to have to move towards side 2. This water level will increase, this water level will decrease, and eventually they will be equal. But what if we have solute? And solute's really anything that's dissolved in the water, any solid that's dissolved in the water. So this could be salt, it could be sugar, anything that's dissolved in the water. When we're talking about osmosis and passive transport, we care about concentration, not water levels. We don't care about the level of water. We care about how concentrated or dilute the solute is. Now, if they're concentrated, that means that the dots are really close together, that they're packed in there, they're concentrated, or there's a lot of them in close proximity. If they're dilute, that means that they're spaced pretty far apart, that there's so much water in between them that they are allowed to move around freely and they rarely come in contact with each other. So if we want to have equal concentrations, in other words, equal spaces between the dots, and the only thing that can move is the blue water, we would actually be moving from side 1 to side 2. Now, why is that? Well, if we take water away from here, it's going to make these dots come closer together. If we put more water on this side, it's going to spread these dots farther apart. We're concerned about the space between the dots, so by making these closer together and these farther apart, we will reach our equilibrium. So in order to achieve equilibrium, water is going to move from side 1 to side 2. This is going to come into play when we talk about isotonic, hypotonic, and hypertonic solutions. In an isotonic solution, the solution surrounding a cell has an equal concentration as the inside of the cell. So the solution surrounding a cell has an equal concentration as the inside of the cell. So if we have a cell floating in solution, we have a concentration out here and it's equal to the concentration inside of the cell. In an isotonic environment, there's no net movement of water, so water kind of flows in and out at the same rate. In a hypotonic solution, the solution has less or a lower concentration than inside of the cell. So what happens in this situation, and I'll draw hypotonic over here, we have this water level, we have our cell. Hypotonic solution looks like this, and then we have all this concentrated stuff on the inside of the cell. In a hypotonic solution, we are going to want to dilute out the inside to match the concentration of the external environment. So water is going to want to rush into the cell, and that's why this cell looks swollen. Now, this is okay to a point, but if too much water rushes into the cell, this cell could burst open. So we have to be careful with hypotonic solutions. If you go to the hospital and you get IV fluids, they're not going to give you distilled water. Because if they gave you distilled water, it would create a hypotonic environment, and eventually you could have something called hyponatremia, or dilution of the sodium in the bloodstream, or eventually hemolysis. Your blood cells could rupture, and that would be really bad. What they do in a hospital is they give you a saline solution, so a salt solution. They're trying to give you an isotonic solution to increase your overall fluid volume without affecting the shape of your cells. So that's hypotonic. Hypotonic, the solution surrounding a cell has less solute than the inside of the cell. So you'll have a net movement of water into the cell. Now in a hypertonic environment, hyper means more or higher. So in a hypertonic environment, the solution surrounding a cell has a higher concentration than the inside of the cell. So water is going to want to rush out of the cell to dilute out the surroundings. This is kind of what happens when you over-fertilize a plant. All of that solute, the fertilizer, is going to draw water out of the plant. So in order to fix that, you have to provide the plant with a lot of water, and then it's going to be beneficial to the plant. But in a hypertonic environment, water rushes out of the cell, and the cell is going to shrivel. So those are the different examples of osmosis. So now we go on to facilitated diffusion. If you guys want to leave the classroom, are you able to just float through the walls of the classroom, or do you have to go through a doorway? Well, obviously you have to go through a door. Think about facilitated diffusion as a doorway. Facilitated diffusion involves a protein or a transport protein. Kind of like a doorway. Now I talked about how molecules can't just go through the membrane. Unless they're small lipid molecules or nonpolar molecules, they can't just float through. So how do we get substances through the membrane when it's so selectively permeable? Well, we need these transport proteins. Facilitated diffusion is still passive transport. It does not require energy. But there's two different types of facilitated diffusion. There's channel-mediated and carrier-mediated. Let's say you guys are all waiting outside in the hallway for class to start. I open the door and you all just walk through the doorway. You go from an area of high concentration, so where there's a lot of you outside, to an area of lower concentration, where there were no students inside of the classroom. That's channel-mediated. So channel-mediated, it's kind of like opening a doorway. But this channel is specific to what can pass through. So we have sodium channels, potassium channels, chloride channels, glucose channels. So they're very specific in what can pass through. Just like my door is specific to only allowing students through that are registered in the class. Carrier-mediated is a little bit different. Through carrier-mediated, imagine that I'm standing in the doorway. I'm physically grabbing onto your arm and shoving you through the doorway. That would be carrier-mediated and probably illegal, and you probably wouldn't want to take this class anymore. But with carrier-mediated, the protein channel actually binds to the molecule that it's about to pass through. So there's an interaction there where it's not just an open doorway where they flow freely. They actually carry the substance through the membrane. So those are the two major types of facilitated diffusion. Either way, they involve a transport protein, and they're still going down their concentration gradient, so it's passive transport. It does not require energy. Now if we go to active transport does require energy. It's the movement of substances against their concentration gradient. From an area of low concentration to high. And that requires energy. These molecules don't like to be packed in. They like to have equilibriums on both sides of the membrane. Especially if they're charged molecules, like ions, if they all have the same charge, they're going to be really upset being next to each other. But we need to have this happen because in order to have the electrical conduction system of a nerve impulse, we need to set the stage for that by having different charges on different sides of the membrane. So for the example of a neuron, when we have a nerve impulse go on, we have an overall positive charge on the outside of the neuron and an overall negative charge on the inside of the neuron. And the exchange of charge across the membrane allows us to generate a nerve impulse. So we have to have a way to set the stage for that. So this requires energy. Packing all of these on one side of the membrane definitely requires energy. Now, other types of active transport, these require energy, known as bulk transport, are listed here. Exocytosis is the movement of a large substance outside of the cell. So here's a picture of exocytosis. Let's say we have something that we want to get out of the cell. Let's say this cell just ate a bacterium and we've digested it using a lysosome, and now we want to get this stuff out of the cell. Well, the vesicle would fuse with the cell membrane, and then eventually just everything would come out of the cell. So through this fusion, through the exocytosis process, those substances would be eliminated from the cell. Endocytosis is bringing a large substance into the cell. Now we have phagocytosis, which is cell eating, and pinocytosis, which is cell drinking. So with phagocytosis, we're bringing a solid substance into the cell. With pinocytosis, we're bringing a liquid into the cell. Here's a picture of endocytosis, particularly phagocytosis, so when we're bringing a solid substance into the cell. So what happens is this substance that's supposed to come into the cell kind of bows in on the membrane. Eventually the membrane pinches off and becomes a vesicle that's freely floating in the cytosol of the cell. Usually there's special receptors on the surface of the cell membrane that these bind to in order to stimulate this process to happen. But this is endocytosis. Endocytosis is bringing a large substance into the cell. If it's solid, we call it phagocytosis, or cell eating. And if it's liquid, we call it pinocytosis, or cell drinking. So that's it for this week's lectures. Make sure you complete the study guide, and I'll see you guys in class.